And we're going to start the second half of this morning's session with another keynote speaker. This is Michael Roukes, who is a professor of physics, applied physics, and bioengineering, and was the founding director of Caltech's Kavli, is that how you say it? Nanoscience Institute. He's going to speak to us today about single molecule and single cell sensing with nanoelectromechanical systems. Well, you're scheduled for 45 minutes, but it's interesting enough. You can talk as much as you like.
It's impossible to dim the lights a little bit.
Yeah, can we dim the lights a little bit? There you go. Perfect.
Thanks a lot. It's a pleasure to be here and to see some old friends. I thought I would tell you a little bit about some work that we've been doing for, it's like going on 14 years, I think now, trying to take promising nano devices and turn them into systems that can actually be useful for biological measurements. And the 14 years maybe in itself is a sobering tale about what's involved in trying to go from some concept of devices and initial papers where you say this has great promise for doing interesting things to actually doing interesting things. And so we're starting just to get to the phase of doing interesting things, as I'll try to convince you. So the story starts with an ultra-high frequency nanoelectromechanical systems resonator. There's a picture of it here. The thing that's down at the bottom labeled C is a doubly clamped beam that's suspended above a silicon substrate. And if you make measurements of the resonance of this beam electrically, in this particular case, this was read out by magnetomotive transduction. I can go into details in question period if you're interested in that kind of stuff. The resonant frequency for this little device is just below 429 megahertz. You can see the amplitude and the phase of the resonance. So think of this as like a little guitar string that vibrates at ultra-high frequencies. In order to get good measurements, we had to devise over the course of those years I told you about ways of suppressing background, because often when you try to first measure nano devices, background is all you measure. So after a bunch of time, we got pretty good at it, and we could look at the resonant frequency of this device. That's what's shown here as a function of time using a phase lock loop. So we could read out, have it continuously vibrating, continuously driven in phase lock, and look at frequency fluctuations. And here it shows you the frequency fluctuations of this thing on this time scale are about a part in 10 to the 7, which we worked hard to get and we're very proud of. But any frequency control people who are used to big silicon devices would think this is totally pathetic, because good frequency control is a part in 10 to the 14 or so. Nonetheless, these are incredibly sensitive devices, as I'll show you, and this is good enough to get atomic scale mass measurements. If there's time, I put in a couple of slides at the end to come back to this question about why are nano things so noisy. So we'll see. Basically because they're small if I don't get to it, but I'll tell you more about it later. So we decided in 1999 that one of the interesting things to do with these little nanomechanical resonators was to stick atoms to them. And we did some initial back of the envelope estimates, and we convinced ourselves that in fact when you stick atoms or molecules on there, you should be able to sense them. And I won't go through all the history of sort of building up to the sensitivity that we have today, but the bottom line is that in around 2006, 2007, we got our first data where we could actually see individual molecular absorption events in frequency shifts of these devices. So the black is in the absence of a flux of molecules, and when we turn on a flux of molecules of bovine serum albumin, which is 66 kilodaltons, or roughly 66,000 AMU, we see every time a molecule accretes onto the device, and it sticks because we keep the device cold, just like frost on a window pane, physisorption. Every time a molecule accretes on the device, we see a frequency shift downward because the resonant frequency, you know from freshman physics, goes something like the square root of the stiffness over the mass, and if you increase the mass, then the frequency has to go down. These are the guys that did it. So the principle of this mass sensing is then fairly straightforward. We have a resonant response. We have, for this little sort of guitar string that's oscillating at high megahertz range. Sorry about that. When my wife rings me, there's a beautiful harp playing and everything else is a quacking duck. When the molecules accrete onto the device, as I said, the frequency has to go downward. And we can think of this sort of formally in terms of an equation where the output is the frequency shift, the input is this little change of mass, the accretion from one atomic species or molecular species onto the device, and it's proportional in a certain regime to something we think of like a gain or a responsivity more formally. This added mass times this mass responsivity gives us a frequency shift. Okay? And here you can see in real time for these data, background fluctuations, nothing's accreted yet. And when the first device comes in, this whole thing rescales, and you can see there's the first atomic absorption event. Okay. Now, how good can this actually be? How far can we push it? Well, back in early 1999, 2000s, trying to look ahead and see what the date was of this paper. I forgot when we published it. But basically, we looked out in the literature and asked what are the ultimate limits, the ultimate physical limits for doing these kinds of mass measurements. And we didn't find any, so we set off to actually do an analysis ourselves and essentially developed this formalism and an understanding that the minimum resolvable mass is inversely proportional to this mass responsivity times noise, frequency fluctuation noise in the problem, because the frequency instability masks our ability to see those jumps downward as a function of time when atomic species accrete. And so this paper explores a number of the fundamental limits to mass sensing, and I'll come back to this at the end of the talk again if there's time. But we convinced ourselves that it should be possible to resolve basically the accretion of protons onto the device. So 1 AMU mass sensitivity looked quite feasible to us in the early 2000s. And so given that, this looks like a really interesting prospect for turning this into a new type of mass spectrometry. Okay? So how do you minimize the resolvable mass? Well, you want to make this term in here, the mass responsivity, as big as possible, so the inverse basically gives us something that's small here, namely the mass resolution goes down as far as possible. And the good news is that for nanoscale, for all types of devices, the mass responsivity scales basically inversely proportional to, if you scale all dimensions down simultaneously to a small device, scales inversely proportional to 1 over length to the 4th power. So what this means is, even from going to micro to nano, this L to the 4th will give you 12 orders of magnitude improvement. Okay? Just the 3 orders of magnitude in size and going from NEMS to NEMS makes all the difference. And so, our early experiments actually proved that this was true. Going from quartz crystal microbalances with mass resolutions in the microgram range down to nanomechanical devices, now with mass resolution in the octogram range, 10 to the minus 24 grams. And in fact, our work with top-down nanofabricated devices, basically has sort of peaked back in the mid-2000s with zeptogram or slightly sub-zeptogram nanomechanical mass sensitivity, and work that has been done in several places, but the first being in Andreas Bachtolt's group in Spain,
showed that with a single low carbon nanotube, they could get down to this 10 to the minus 24 grams. Now this is, sort of represents the truth in what we've been saying, that you can get these 12 orders of magnitude improvement, and it looks like you can get down to single AMU or single octogram mass resolution, pick your favorite units. But the next step is to apply this into some useful technique. And so we know, and I'll come back to this again at the end, we know that macromolecules that we want to measure are many times larger than the width of these devices. And so these are not useful devices. They're useful devices if you want to look at atomic species, like low mass atomic species. But if you want to do biological mass spectrometry, we need to stick with basically CMOS compatible technologies, doing top-down and using widths that are roughly no smaller than the size of the molecules themselves. I'll come back to that. So how do we turn, this sort of nanophysics demonstration of, hugely sensitive mass sensing into mass spectrometry? Well, just let me remind you what mass spectrometry is. Mass spectrometry, we start with biological or chemical analytes in the liquid phase, and we spray them into a high vacuum situation in such a way that ultimately the species that are, the ionic species that are dissolved in solution ultimately lose their solvent phase, their aqueous phase, and by the time they get to the mass analyzer, are denuded of all this solvent or aqueous phase, and they're essentially bare macromolecular ions. These ions can then be manipulated in magnetic fields or electric fields. Okay, so we ionize these species that are in solution, we denude them of the solvent, and then we can basically transport them into some sort of either electric or magnetic or combined mass analyzer that separates them out into packets that are delayed in time, that have different mass to charge ratio. And these different packets of ions that have different mass to charge ratio then are delivered at different times, and we detect them as blips in charge deposition onto a charge detector. Okay, and through that, and through something like 50 years of excuse me, difficult, hard-won technology to convert mass to charge ratio into biologically useful information, people have developed this tool, which is now the mainstay for proteomics, okay, which is proteins, as you know, are the machines, life's machines, basically. So, what's the difference between this previous paradigm and nanomechanical mass spectrometry? Well, nanomechanical mass spectrometry is actually quite a bit simpler conceptually. We start with, again, preparing our species in the aqueous phase, injecting them into vacuum. Now, in this case, we don't actually need to have ionic species because we're not going to detect charge. We're going to detect the inertial mass of the species. We're going to spray these onto a pixel array where each pixel is an individual mass detector. It's going to register clicks with the incoming atoms. And then we have some sort of highly multiplexed electronic readout. Now, where is the mass analyzer? Remember, there was a mass analyzer that separated the spray of molecules into different mass to charge ratios. Well, we don't need that because our mass detector is the mass analyzer. We can read out the frequency shift is directly proportional to the mass of the accreted species. So NEMS-MS acts as both the mass analyzer and the mass detector. Advantages are, as opposed to conventional mass spectrometry needing millions of molecules, we can do single molecule detection. Single Dalton resolution has been shown for atomic species, and we believe it's still possible for macromolecular species. We can measure extremely large masses. This is key if you want to look at protein complexes, for example. Big assembly of machines, for example, ion channels and things like that. I mean, there's many, many examples. Ribosomes, for example, others. These are, in some sense, on the scale of the forces that are involved in ionization for conventional mass spectrometry, ionization and going into the fluid phase. Typically, large species are weakly bound on sort of Kolubek scale and basically break up. And so it's difficult to do macromolecular complex mass spectrometry with existing paradigms of mass spectrometry. Whereas we can do this with neutral species, and we can look at extremely large species without loss of resolution. In fact, we have more and more resolution as we go up in mass. We can make many measurements per pixel, so that we can, if we actually put thousands of mass spectrometers in a chip-based paradigm, we can actually get high throughput detection. But of course, all of these things that take the initial demonstrations that we've done with a single nano device and scaling it up to biological utility require all the tools for semiconductor large-scale integration. And to this end, over the past 6 years with collaborators in Grenoble at the equivalent of the Department of Energy's Laboratory for Electronics and Information Technology in Grenoble, also Minitech laboratory there. We've developed standard NEMS process. We developed NEMS-CMOS processes and 3D NEMS integration processes to allow this kind of large-scale integration for these mass detectors to occur. Now, a complication arises, and we saw this basically in our first paper where we measured these single molecule jumps for accretion, is that, and this wasn't unexpected, that when we went to larger molecules, this thing that I'm going to tell you about became extremely manifest. That is, even though we were sending in a single species, in this case beta amylase of a few hundred kilodaltons, we got jumps of different height. Now the reason why we got jumps of different height is because there's a position dependence of the mass sensitivity. If you think about it, if you're operating this thing in fundamental mode, there's antinodes here, there's nodes here and an antinode in the center, so the maximum of the vibration is at the center, and you would expect where the thing is vibrating the most, you actually have the biggest effect on the inertial mass when you stick a little species on there. So how do we get around that? So how we get around that is, it turns out that each of the modes of the nanomechanical device, the first one being this, the second one being this, and so on, depend differently on the position of the accreted species. So in simplest form, we can actually, instead of just applying one control loop, we apply two of these low noise control loops to simultaneously drive two modes, in this case the first and the second mode, and read out, so here's the first and the second mode, and read out the time correlated jumps in the frequency that occur with the accretion of an individual species onto these devices. Notice that all the jumps occur in a time correlated fashion. They're happening to the same device, but they affect the two modes differently. Okay, so now we have two measurements, and we have two equations for mass and position unknowns, and we can basically take these two position-dependent frequency jump equations and solve them to actually get both the position of the accretion and the mass of the species in real time for each molecule that comes into the nanomechanical device. Okay? So when I say we're making arrays of hundreds or thousands of mass pixels that are coupled with CMOS electronics, each of these pixels has to be a multi-mode detection, right? Because we want to, when a molecule comes in, we want to capture it effectively. There's a plume, right, that comes out from our spray. We need to tile the area of that plume with good filling fraction, with lots of nanomechanical devices. Each one of them is a pixel that's read out independently with this multi-mode detection. And that's what we've been building with our colleagues in France. Now what's interesting is that with this multi-mode detection, for part of his PhD thesis, my former grad student, he's now a member of professional staff at Caltech, Salim Haneh, basically understood that we could evaluate the analyte's uncertainty in mass and position of absorption with knowledge of basically the measurements of the noise sources in this process. So we can go from the experimental data, which is, as a function of time, the time-correlated frequency jumps associated with these individual accretion events, to translating these things into essentially, here's the frequency jumps now. So the frequency jump plane, and now it turns out that analytically we can calculate what the contours are of constant mass and constant position, and then plot essentially out the probability distribution function in the mass and position plane. So each one of these is now like a little spectrum, if you will, both in position and in mass, with uncertainty associated with the frequency uncertainty or the noise uncertainty in our system. Now let me just take up, before I show you some recent data that we've gotten with this system. It turns out that, let me tell you a little bit about where we've been going with actually the front end of the system, trying to make it more and more amenable to doing useful biological experiments. The first thing that we used for injecting our protein species into these systems with the nanomechanical device at the end was electrospray ionization, which is a standard process that's used to get in mass spectrometry with a charged needle to essentially get protein ions in solution to become denuded protein ions in vacuum. Okay, so usually you inject in, with an electrically charged needle into the vacuum space, and then you go through differential stages of vacuum to progressively evaporate away the solvent. And we transport this in sort of a coherent, spatially localized way by using radio frequency ion guides, hexapole ion guides, that ultimately deliver the molecules down to the surface of our individual nanomechanical device, or now into our nanomechanical arrays. So we electrospray the solution, droplets enter the capillary tube, okay? Droplets in the system here now undergo Coulombic fission. Ultimately only a single protein ions remain, and then the proteins are guided down to where our nanomechanical device is. In our first systems, we were going from the era of making measurements at low temperature, and high magnetic fields for mesoscopic physics. So, I converted, with my postdoc, converted one of our previous systems into our first mass spec system, and our delivery distance was like a couple of meters. So you can imagine our efficiency was really small. These are now been converted to tabletop systems. Now, what we've been going to lately is a variant of what's called MALDI, for those of you who are familiar with mass spectrometry, which is matrix-assisted laser-induced or laser desorption ionization. Laser desorption ionization. We're going from there to a sort of a new variant, which is called laser-induced acoustic desorption. And the idea here is, on the front surface of a plate, you have the species, or these can actually be individual cells that you're interested in analyzing. So either macromolecular species, proteins, or individual cells on the front surface. And the back surface, basically you blast with a laser in a spatially accurate way. This is a multi-layer structure that's designed so that the optical radiation that hits on the back is converted to a shock wave, an acoustic shock wave, and that propagates quickly to the surface and then blasts off the molecules or the cells into a little plume, and now we put at close proximity our pixel array of mass sensors. Okay? And of course we did this initially with a single mass sensor that's shown here. So proteins are deposited on the substrate, and this is basically the same process that's used for preparing MALDI plates today for conventional mass spectrometry. The substrate's loaded into a UHV chamber, a pulsed UV laser hits the substrate backside, and then the shock waves happen. Proteins dissolve, and then physisorb onto a nearby cooled NEMS array, and we can detect their mass molecule by molecule. So one of the first things we looked at are antibodies that occur in human blood serum. These are called immunoglobulin M's. And immunoglobulin M's are sort of a Y-shaped molecule And important for the story that I'm going to tell you, and sort of leading up to thinking about looking at protein complexes, these things in blood serum are most prevalent in a pentameric form. They sort of associate into a biologically active pentameric form. But there are other isoforms. The pentamer is one isomer, isoform, hexamer, tetramer, a whole bunch of different isoforms occur. And in fact, these isoforms have different biological or immunological activity. If you have zillions of molecules, you can run a gel electrophoresis process and actually see different bands under different conditions for these different isoforms. Okay, again, this is having jillions of molecules. But if you only have a few molecules, you can use NEMS-MS spectrometry. And so this shows 73 events for detection of this aqueous phase, this human serum IgM. And the way this is built up is that here's the first event that happens. Okay, the second event that happens is here, and we carry along for a ride in this representation of the first event. The third event happens here. This is the first and the second event, and so on, right? And so we can build up slowly, molecule by molecule, the conventional spectrum that you get for IgM in solution. Okay, and you can see basically all these different prevalences based on the mass of the species. So like 1 megadalton is the pentameric form. Now usually all you get in mass spectrometry is the white trace, right? But we get these individual units one by one, and so we know deterministically how this white trace is actually composed, okay? Again, usually you get the sum curve when you do mass spectrometry. We get these individual units. So this is in some sense a reversal of the traditional situation where in the traditional situation you get the mass spectrum, you postulate what the individual units look like, and then you use a multi-parameter fit to actually try to figure out what their spectral weights are. In this case, our measurement gives us directly the spectral weights. We know for this particular experiment, we got precisely 7 molecules of trimer, 12 molecules of tetramer, 20 molecules of pentamer, and so on. So this is sort of an exciting new development for actually looking at the prevalences in a quantitative way for these different species. So where would we like to go next with this technology? Well, remember I said that we're working with devices that are larger than nanotubes, and we're forced, and that nanotubes give you single AMU resolution, which allows you to look at protein fragments in the standard realm of biological mass spectrometry and begin to figure out what's what. Right now, with our mass resolution floor being something like 20 kilodaltons, our principal area of application has been proteins and large proteins, protein complexes, viruses, and as I'll tell you about, some work with individual bacterial cells. So what's interesting in this range that we can access is the first question. How can we maximize the efficiency and throughput? So in this case, the question arises from the fact that all of the initial measurements that we made were made with individual nanodevices. And if you think relative cross-sectional area of the plumes that we inject, that we deliver, compared to the capture cross-section of the individual nanodevices, it's infinitesimal compared to the whole. So our efficiency is really small. It's like a part in 10 to the 9. For every billion molecules we inject, we collect 1. I often get the question, how can you look at one by one, how fast do the molecules come? Well, if you have a capture efficiency of 10 to the minus 9, the molecules come every minute or something like that. So, why do we have traces with 73 molecules? Well, that's about as long as we want to make the run, right, to capture those 73 molecules. Clearly, that's inefficient. It's a demonstration. We need to figure out how to get millions of molecules per second. So what is the roadmap for actually doing that? And we're well into that, as I'll show you. And then finally, the question of, if we stick with devices that are large enough to be useful biologically to capture biomolecules, not individual atomic species like nanotubes, but things that have appreciable cross sections, like maybe 50 nanometers or so, 100 nanometers at the outside, can we push down the mass resolution to get single AMU? And back in this paper that we wrote in 2004, we convinced ourselves that that was possible. Why haven't we achieved it? So these are the three things that I'll talk to you about. So targeted near-term applications first. So I talked about macromolecular complexes, and one of the things that we've been studying, but I won't go into the details of our results so far, is things like ribosomes that are composed of many different protein subunits, right? And it's interesting to actually look at, for example, samples that are taken from inside the cell, where one can look at the state of completely assembled ribosomes and actually all sorts of other like misformed composites and things. So we can actually look at, on a single cell level, begin to understand, through incompletely formed complexes, what are some of the processes by which this macromolecular assembly is carried out. Another example is that recently there have been advances in conventional mass spectrometry where it's become possible to do fingerprint analysis on bacteria, not on individual bacteria, but on cultured bacteria, where you have billions of bacteria, you grind them into a homogenate, and if they're all one and the same bacteria, you can get a mass spectrum fingerprint of protein fragments, and in this case it's between 2 and 20 kilodaltons, of protein fragments with the bacteria, and it's been found that there are characteristic fingerprints, these mass spectra between 2 and 20 kilodaltons for bacteria, that are sufficient to identify bacterial species. Now this is interesting because bacterial identification is key for early stage application of antibiotics for these strongly resistant strains of bacteria that give people sepsis in hospitals, and is a significant worry because if you apply all sorts of antibiotics when you're before you figured out what it is, and you've got to do it quickly before the person basically becomes deceased. If you apply all sorts of antibiotics, basically you'll kill off all the other ones except for the resistant one, and then that one will even more accelerate its deleterious processes in the body. So you really need to nail sepsis by having an early stage diagnosis of what bacterium it is, and then have a quick target for those specific bacteria. But again, this is not something that we have today. It's an extremely quick identification of bacteria. So we'd like to take this idea of bacterial identification down to the single bacterium scale. We don't have to wait 2 days to do the culture because the patient might be dead, but actually do it basically instantaneous in real time. And so we can translate our technique into a single bacterium model, that will actually do the job. I'll say a little bit more about that. Another interesting thing in the vein of what I was talking about with ribosomes is actually looking at assembly processes for viruses. And just like chemical reactions, if one can actually look at all the variants of chemical processes, things that actually don't go the right way, we can actually begin to understand different processes that are taken in an assembly process. And so we need to have sort of single species resolution in order to do that. How can we maximize efficiency and throughput? How can we get from 10 to the minus 9 efficiency up to at least the few percent efficiency that conventional mass spectrometry has, and perhaps even better? Well, again, it's by tiling this area that we inject the plume with densely with NEMS-MS pixels, and that's going to require large-scale integration of nanomechanical devices. So back in 2007, with our colleagues in Grenoble, we transferred our CMOS-compatible NEMS processes in and into the foundry, and worked out all the bugs in getting this to work on the 200 millimeter 8-inch wafer scale and develop nanomechanical devices. And then since then, obviously we need per pixel a fair amount of electronics power. We've co-integrated this with CMOS, and we're beginning to do 3D stacking runs to figure out how to do this 3D integration. And the reason for the 3D integration is perhaps you know how the Texas Instrument micromirror works for digital projectors. It's basically a dense tiling of little torsional devices, and these devices have to densely tile so you have good optical coverage, and below them is all the electronics that drives the individual pixels. And so we're essentially attempting to do at the nanoscale the same sort of things with these multimode mass sensors. So what about trying to get down from 20 kilodaltons and above, this realm that we can study protein complexes in large protein macromolecules, down into the few Dalton range, which is the province of looking at protein fragments and allowing us to do quantitative mass spectrometry. How can we actually get there? And again, the first thing to say is that we need devices with the size of macromolecules, so we can't take a shortcut and use nanotubes to get there. We need to stick with these CMOS compatible, again for the large-scale integration aspect, but also because of the size, needing to be comparable in size of macromolecules. So what are prospects for improving mass resolution if we restrict ourselves to the VLSI platform and top-down NEMS devices that are 50 or 100 nanometers in size? Well, the first thing is, how do we get our minimum resolution down from 10 or 20 kilodaltons to a few AMU? I mentioned that the first paper that we wrote said that would be technically feasible, and we've spent about 10 years exploring how close we can get to fundamental thermodynamic limits with this mass resolution. And the bottom line is that there right now is what we perceive to be an engineering brick wall. There are noise fluctuation processes that we don't completely understand, that are probably surface mediated, that we've been exploring, but this is complicated, difficult surface science that needs to be applied to nanomechanical devices. And so we're continuing to investigate that. Now, in our defense, I'll say, if defense is needed, that even in large macro devices, we don't really understand in great detail at the microscopic level what's the origin of noise processes. And so maybe it's a bit arrogant or brash to think that in a short period of time we'll understand these for nanomechanical devices, but you can see the reason why this is really imperative for us. So we continue to push on that. We believe that we have advances in hand to allow us to go down to something like several hundred kilodaltons within the next couple of years. Now, a second thing that actually goes into the mass sensitivity is basically how hard you actually drive the devices. Those of you that know about making oscillators, you know that you get the best phase noise performance, the purest tones out of oscillators if you drive them extremely hard, all the way up into some sort of natural limit. And it turns out that that natural limit of driving is typically the nonlinear regime. Once you go beyond once you go into nonlinearity, you start making harmonics, and the amplitude of the system depends on the frequency that you're at. And so if you have frequency fluctuation, that's converted to amplitude fluctuations and vice versa. And so we've looked at this very carefully, and instead of just assuming that this conventional paradigm is true, namely that if you want to make good oscillators, which is what we're doing with these pixels, if you want to make good oscillators, you keep them in the linear regime. We've looked at that, and we've found that, in fact, we can go strongly into the nonlinear regime where we have this amplitude to phase noise conversion. But we can find special this is a paper that just came out in Physical Letters. We can find with these nanomechanical oscillators that are based on nanometer resonators. We can find special places in the operating curve where, in fact, this amplitude to frequency noise conversion is suppressed. So this is another way that we're exploring, and this in part will help us to actually push down the ultimate limits of measurement. So let me just conclude with a couple of slides, and I think there'll be enough time for some questions if people have them. Part of our roadmap right now is trying to push this and see if we can do this single bacterium identification for mass spectrometry. But what we'd really love to do is using microfluidic systems that are well developed, and you know many great examples from laboratories here of single cell handling of microfluidics, to be able to do mass spectrometry on a cell by cell level, to understand, to take snapshots instantaneously of what all the machines in the cell are actually doing. And just to give you some sort of numbers for this, with 10,000 analyzers in parallel, which is a medium scale integration feat that we think we should be able to do, with a protein flux that we can handle now of about per pixel of about 1,000 molecules per second, we should be able in less than 2 minutes to measure essentially all the proteins, the billion proteins in a cell, and then go through many, many cells with, let's say, arrays of mass spectrometers that are individual boxes, made into some sort of a screening laboratory. I was going to say something about imaging mass spectrometry, but I think I'll skip that in the interest of allowing people to ask questions. So we've been pushing recently towards, after a bunch of years of just sort of doing the physics of mass sensing and understanding noise in nanomechanical systems. Since about 2006, we've been really pushing to try to develop systems using these for doing biologically interesting mass spectrometry. And it's going to take us a few more years to get where we want to go. But I think it's an interesting and worthy enterprise for actually taking these nano devices and turning them into nanosystems. And here's all the great people that I've been privileged to work with on this project and some of the funding that's actually supported this. Thank you for inviting me to speak to you.
As before, we're going to take a few questions, but first I'd like to present Michael with this plaque and our appreciation for coming out here to visit us and present this keynote address this morning. So
Thanks very much.
Thank you very much. Time for some questions.
You mentioned about the use of non-linearity.
Yeah.
Have you tried parametric explanations?
So we have parametric. So we've done an extensive amount of work with parametric mechanical systems, and parametric processes don't improve the fundamental signal-to-noise ratio that you have. But in going from the mechanical domain to the electrical domain, they can basically boost the displacement amplitude of the system and allow you, so it doesn't improve the signal to noise at the front end, but if your signal to noise in large part is limited by displacement transducer noise, it can give you big displacement to overcome that noise. So it turns out in these systems, our displacement transducer noise is not the principal thing that's limiting this. It's basically this anomalous phase noise that I was alluding to in the mechanical domain. And for that, we need to get into the mechanical, we need to do all of our engineering in the mechanical domain to really understand what the origin in the mechanical domain is of these processes. So I'm not, and we can talk more about parametric stuff, but with the specific context of this discussion in mass spectrometry, parametric won't really help us because we've already done a good job of engineering the noise in our transducers.
So moving toward your single cell goals, how do you do your on-chip sample processing and more speaking of salts and fuel salts
So this is work that's in progress, and we haven't worked out all the details about what, in terms of the salts, are important in actually getting these mass spectra. But all the sample preparation that we've been using, so we have partners from Bioengineering, which is a big pharmaceutical company, sort of a parallel Becton Dickinson in the US, that does these biological fingerprinting using mass spectrometry, we're using the same protocols that they have. It's not really an answer that I can talk about in
What is your
being intentionally evasive. Full disclosure.
what's your experience with functionalizing these, and would you think that could improve efficiency?
So remember that we're doing everything in the vacuum phase. And molecular recognition in the fluid phase is based on secondary, tertiary, quaternary structure of proteins. And as soon as you absorb them onto the surface of the device, if it's in fluid, you can preserve much of that three-dimensional structure and preserve the molecular recognition lock and key process. But in vacuum, the proteins denature, you lose all that. So you can't make these biologically specific because we're working in the vacuum phase. So that's why we rely on either microfluidic preprocessing, you can use immunospecific microfluidic preprocessing upstream from the detection in the same way that you do, cell-D and other things, in mass spectrometry, or at the downstream end, at the NEMS themselves, we look at, exactly the inertial mass of the species as our discriminator.
So I know that your group has also done nanomechanical resonators with nanofluidic channels built in.
No, we haven't.
Oh, you haven't?
No, we've been collaborating with Scott Manalis, but we haven't ourselves done it.
Right. I saw a paper nano letters.
Yeah.
So is there what are the limitations of that technology? When you actually put it all together?
So you could do what was previously described about using immunospecific detection in small channels for fluidic detection with high quality factors. So for those who not fully apprised of what's being alluded to here, when you take, the reason why we get these 12 orders of magnitude improvement is that we stay in vacuum. We preserve high Q for the devices. And the question is, if we take these into liquid phase, where we can do some of this immunospecific molecular recognition for specific species directly in the liquid phase, how well will they work? But we still get this incredible mass sensitivity. Well, what happens is that they get fluid bands. The quality factor goes down. You get 4 or 5 orders of magnitude decrease in the mass sensitivity. So, our colleague Scott Manalis and his group at MIT said, well, let's not put the devices in solution. Let's put the solution in the devices and keep the devices in vacuum with little fluid channels inside, and then you can do all the fluidic stuff into these little micro or nano channels. Now, so what we've done, our contribution, is actually looking at how far you can scale these fluid embedded nanochannels, their sensitivity for immunospecific detection. And the big problem comes, so they end up doing pretty well, comparable to other sensors, but because of a whole bunch of technical difficulties, you never really get the full promise of what we can do in vacuum. And one of the big limitations turns out to be that the throughput in these little nanochannels means that unless you make arrays of thousands of these individual nanochannel resonators, you don't get sufficient throughput to look at biologically useful quantities of fluid to do analyses. When you're talking about, for example, looking at HIV detection and trying to do it without PCR, for example, you're up against a really stringent criterion of only looking for a couple of molecules per milliliter, right? And then you think about non-specific binding and how much fluid you have to push through in a milliliter, but it becomes prohibitive to actually, even though it's in principle feasible, practically there's a whole bunch of reasons why we can't do it.
We should probably move on, but we won't let Jim leave town until you have a chance to talk to him at lunchtime. And thank our speaker again.